So what I will be talking about is the role of alternative splicing in cancer, but not so much in cancer as in tumor progression. Because cancer is bad, but what is bad of cancer is the capacity to spread in your tissues. You all know that it is known that there are lots of genes that have an altered splicing pattern in cancer, but the difficult thing is to distinguish between the cause and the effect. Okay? So, it's cancer that causes altered splicing pattern of many genes, and that for sure happens. Or is the altered splicing pattern of some specific genes that causes cancer or progression of cancer? So that's the big question. All these issues are discussed in a review that I have sent to the course organizers as teaching material. I don't know if you have got it. You haven't got it? You should get it on a on a CD. Anyway, so there are many genes that have an altered splicing pattern. The most famous one is BRCA1, in which exon 18 is skipped, and its functional impact is higher susceptibility to breast cancer. And you have heard of this actress that has removed his breast because she has this mutation in BRCA1. What I will be talking about instead, and then, for example, this one is another famous case, CD44. And this is bad because the inclusion of these two exons, or these two exons, or this one, leads to the acquisition of metastatic potential, and so on. But please, read that review and you have an idea of the complexity of the problem. So, as I said, it's difficult to distinguish between the cause and the effect. Because certainly, cancer disrupts everything, including the splicing machinery. And so it's important to, once you have identified an aberrant splicing, it's important to identify the factor that regulates splice selection, to identify the signaling events that modify the activity of the regulators, and to characterize the downstream physiological consequences of the protein that is produced and of its isoform, which are produced by aberrant splicing. And I will present you a specific case in which one single aberrant alternative splicing event has very dramatic consequences. And the case, okay, before you should remember what I already presented you, the fate of the mRNA as after it is transcribed, before going to the cytoplasm to be translated, is covered by this protein, HNRNPs, and SR protein goes through splicing. You remember this exon junction complex that remains bound to the junction site between two exons. So the mature mRNA is exported, it goes to the cytoplasm, is translated, and so on and so on. And the major players in this, all this very complex thing, are the the splicing factor that I already mentioned to you. And please remember this one that has two names, SF2 ASF or SRSF1, because I will talk a lot about this factor. And the HNRNP proteins that I already mentioned, and remember this protein, A1, and also this one, A2B1, A1B2. So there are, this group of protein will come out along. Okay. Now, the alternative splicing I will be talking about concerns a tyrosine kinase receptor for macrophage stimulating protein, and is a member of the scatter factor receptor family. It is involved in control of cell dissociation, motility, invasion of extracellular matrices. So it's a very strange receptor. By the way, the acronym RON is a French one, derives from Récepteur d'origine nantaise. This receptor is involved in cell dissociation, motility, invasion. It's involved also in natural processes like, organogenesis, wound healing. Okay, when you cut your skin, then it heals. And this receptor is involved. Okay, this receptor is an heterodimer, two subunits, alpha and beta subunit, plus all the transducing apparatus that transmits the signal of to the RNA metabolism and so on. Now, this strange receptor has a lot of isoforms produced by alternative splicing. Many of them are transforming proteins. So it's it's a proto-oncogene, okay? And in many cases, the mutation can be truncated, but the mutation that interests us is this one, is a deletion of a very small exon in exon 11. It's called RON delta 165. Don't be confused. This is the molecular weight, 165. This mutation has a consequence that removes a proteolytic site that is cut to produce the heterodimer structure. So two different subunits. In this case, the removal of this tiny exon prevents that, and so and this particular isoform, which is deleted, exon 11, only of 49 amino acids in the beta chain, extracellular domain, is constitutively active in the absence of ligand. So even in the absence of ligand, of the scatter factor that I mentioned you at the beginning, it fires its signal to the cell. It's and it confers invasive phenotype to the recipient cells. How do you know that? Well, we know because many years ago, our colleagues in Turin, at the Institute of Cancer Research in Turin, told us, look, we have many cases of colon and breast cancers in which, strangely enough, this isoform with the deletion of only this tiny exon is overexpressed enormously, please go do something and tell us what's going on. And so we got this suggestion, and we started studying what's happening, what causes the skipping of this tiny exon, and what are the consequences of this skipping. Okay, the receptor is constitutively active in the absence, but what it does, why it is so dangerous. So we studied, we did a lot of work, we published and several papers, and in the end, we discovered that in the exon 12, so in the downstream exon, there is an enhancer of splicing, this one, plus, okay, which when bound by his cognate factor, and the cognate factor is the SR protein SF2, when bound, so when this factor binds the enhancer and is very abundant, being an enhancer, is strengthened this splice site, okay, in comparison to this one, and so the exon is skipped. And you produce the isoform that's called delta-ron. So in the downstream exon, there is an enhancer. Not only there is an enhancer, but there is also a silencer. So I'm summarizing the work of several years and several papers, okay? So there is a regulatory cassette in the downstream exon, which is a bit atypical, because usually the enhancer, it's either in the exon itself or in the intron nearby. In this case, is in the downstream exon. Well, that's the way it is. And what the production of this isoform, what does it do? It changes the nature of the cell. Just this tiny little event, skipping of a small exon, has incredible effects. It induces the epithelial to mesenchymal transition. You know, cells are different types. There are epithelial cells that are in the epithelia, and the epithelia are the tissues where most solid tumor occur, because it's an enormous amount, surfaces, lungs, intestinal epithelial, skin. Epithelial cells have these features, okay? They have sort of, you look at the microscope, they are sort of cobblestone appearance. Okay? Well, if you transform these cells, so if you overinduce ASF, you transform these cells from the epithelial to mesenchymal cells. Mesenchymal cells are the cells that are able to migrate. They don't stay together. They lose adherence, and they can migrate and cross the matrices and so on. So this little event causes a rearrangement of the internal architecture of the cell. So this fact that this protein is constitutively induced transforms epithelial cells to mesenchymal cells. So has the EMT, it's called EMT, epithelium to mesenchymal transition. And there are several indications and markers of this event. And for example, you see the actin is rearranged. E-cadherin, which is an adhering protein, is much decreased if you overexpress SF2, that here is called ASF. Beta-catenin moves, it's not too easy to see it moves, it becomes nuclear, while in epithelial cell is cytoplasmic. And you can measure the capacity of the cells to migrate, for example, to migrate through a membrane, porous membrane. This is the lower side of the membrane that shows that the cells that have undergone the EMT transition can migrate, while the other cannot. So that's what happens. The epithelial to mesenchymal transition is characterized by several features, okay, that I already mentioned. There are proteins and the transcription factor that increase in abundance, protein that decrease in abundance, protein that accumulate in the nucleus, and so on. And this is what happens in a pictorial representation. Okay. You have the tumors, epithelial. If in some of one of the cells you have this transition, the cells can cross out of the tumor, and they can cross all the barriers that they find. They can go into the bloodstream and go elsewhere and give origin to a metastasis. Okay? And this process, which is reversible, you can go back from mesenchymal to epithelial cell, usually occurs at the metastatic front of the tumor, not from inside, but from the surface. Okay? From there, these cells change their feature and start to move. So we did a lot of work. We verified that it's this single event. So I'm not saying that EMT can be induced only by skipping the exon 11 in the RON oncogene. What I'm saying is that this single event is enough by itself to cause EMT. Okay? What was very nice and sort of surprising is that you can reproduce this phenomenon in, these are, of course, SV448 cells are I don't remember which kind of, they are tumor cells, okay, in culture. Colon cancer. Yeah. If you grow these cells at high density or at low density, so the growth at low density simulates EMT. So the growth at low density or high density is a model, is a cell culture model of EMT. But you can see that, for example, E-cadherin almost disappear when cells are grown at low density, and beta-catenin becomes nuclear. It is cytoplasmic, becomes nuclear, and all these other indicators are typical of the transition epithelia to. So this is very nice. So the cell density controls is like EMT, and in fact, the cell density controls the splicing profile of RON and the expression of SF2. So you see, this is RT-PCR. At high density, most of the product is full length. At low density, you have a switch, delta-ron. So here you have the skipping of exon 11. And in parallel, at low density, you have much more SF2 compared to high density cells. So that's you see the correlation. You have more SF2 and more skipping. Here comes the nonsense-mediated decay story. These cartoons represent what Lil McWad has very well illustrated in his lecture. Okay? What is nonsense-mediated decay? Why I'm talking about nonsense-mediated decay? So, well, because turns out that many SR proteins are subject to this process. In other words, this is a paper in Nature, 2007, and it says that many SR genes have an unproductive splicing which is associated with highly conserved and ultra-conserved elements, DNA element. I'll tell you what that means. But in any case, you have, in many SR proteins, you can have a splicing that can occur either in the coding region or in the 3' UTR that produces the isoforms that are degraded by the NMD mechanism. And in particular, so for example, these SR proteins have possibility of having a premature termination codon in the coding region. But what we are interested is this fact. SF2, the guy that controls the splicing of delta-ron, has a premature termination codon here in the 3' delta-ron that is produced by the splicing of this intron, and the splicing of the exon causes the binding of the exon junction complex, and by the mechanism that Lynn has so beautifully explained, you produce this form that is degraded. So the nuclear-mediated nonsense is a way to regulate the protein level of many proteins, including SF2. And this is very interesting, because we discovered that, you remember the model of AMT, cell density, growth, high density. Cell density by itself regulates the splicing, the alternative splicing of SF2. In other words, when you grow cells at high density, you produce, you skip this exon, you produce more form of the RNA that is degraded. And at low density is the opposite. So the density effect, which causes the skipping or the non-skipping of the exon, starts with regulating the concentration of the protein that binds to the enhancer by regulating the degradation through NMD. So this is very interesting. Also because people have looked at the splicing profile of the 3' UTR in normal and colon and other tumor tissues, and they notice that the ratio between full length and the NMD isoform is much higher in most tumors, indicating that a high level of SF2 is tumorigenic. All right? Let's go back to the SF2 story. Say, high level, high density, low density. But not only that. If you take conditioned medium from high density cells, conditioned medium, so you grow the cells at high density in a medium without serum, and you take what the cells secrete, so it comes out of the cells. So the factors, growth factors, cytokines, whatever. And this medium by itself, the content of this medium, is enough to produce the effect on the splicing profile of SF2 3' UTR. In other words, there are soluble factors in the cell conditioned medium that control the UTR, the splicing of SF2. And I don't go through all this. But you can see that a high concentration, you have mostly full-length SF2. If you add conditioning medium, you almost switch completely to the degraded form of the RNA. So what is in the conditioning medium that causes the upregulation or downregulation of SF2? There are growth factors, cytokines, other things. And it is known that all these factors exert their effect by activating protein kinases. Okay? And in particular, this pathway, the mitogen-activated protein kinase cascade, in particular the ERK kinase. And so we asked whether the secreted factor that causes that effect on the SF2 3' UTR splicing, which in turn causes the effect on RON splicing, is discovered that this secreted factor or factors that control the SF2 UTR splicing, it does it through the RAS, RAF, MEK, ERK cascade. So it's a very complicated story, but it's, in essence, what happens is the ERK, which is the kinase that mediates the effect, as you can see, is phosphorylated, that is activated at low growing at low density cells. So low density cells resemble the mesenchymal cells and is not phosphorylated at high density cells. And this has the effects that they have already illustrated. So that is the trick. The splicing of this exon that controls the abundance of SF2, that controls the splicing of RON, depends on something that is activated by the ERK kinase of the SARC. Anyway, And here comes the collaboration with these people in Trieste. Comes another player, SAM68. SAM68, SAM stands for Sarc associated during mitosis. Strange name, but in fact is an RNA binding protein that transduces the signal from growing factors to the RNA metabolism. So it's an RNA binding protein. It does not belong to the category of the SR protein, but it is an RNA binding protein. This is its structure. It has 3 binding domains, which are not the same as the RRM that we've already seen. But most important is the target of the ERK kinase. So these are the amino acid threonine, serine, threonine that are phosphorylated. Okay, so what does this protein have to do with all the story? This protein, so in the SF2 3' UTR, there are strangely 2 ultra-conserved sequences. Next to these ultra-conserved elements, there are 2 binding sites for SAM68, and that's what the spanning was done in collaboration with the people in Trieste. And now we noticed that, or they noticed, these people in Trieste, that there are 2 binding sites for SAM68 in this intron. And we verified, or I say they verified, that indeed SAM68 binds to these 2 binding sites. And then it turns out that SAM68 controls the nonsense-mediated decay of the SF2 transcript. In other words, the binding of SAM68 causes the inclusion of the intron and the production of the full-length protein that is not degraded by the NMD mechanism. So, in other words, SAM68 is, how can you describe it? It produces more SF2. Okay, the activation and the phosphorylation that happens at low density. And here you have all the parameters. Okay, and in fact, if you overexpress SAM68, you make more SF2, and you skip more exon, and you have delta-ron. That has as a consequence the transition from epithelial to mesenchymal. See the different shape with all the markers. Okay, this is the same thing. And the other thing that was noticed that the SAM68 mRNA expression level in human colon and human tissues is, on the average, higher than normal. So the data suggest that the SAM68 expression level or phosphorylation status could play a role in tumorigenesis. But you see the intricacy of the network. SAM68 plays a role in tumorigenesis by affecting SF2 splicing, which in turn affects the splicing of this receptor, delta-ron. So I summarize here. SAM68 phosphorylation is elevated in human breast tumors, tissues and cell lines. And here, if you are interested, there are all the references. SAM68 expression is elevated in human prostate cancer tissues. SAM68 null mice are resistant to tumor and metastasis formation. implicating the role for SAM68 as a proto-oncogene. So SAM68 could contribute to malignant transformation of epithelial cancer by inducing a post-transcriptionally regulated EMT. So what is the model in the end? The model is the following. A low cell density, or at the leading edge of a tumor, for example, that simulates the low cell density condition, the cell put out factors that leads to the phosphorylation and activation of ERK and eventually SAM68. SAM68 binds to SF2, 3' UTR, prevents skipping of the exon and the degradation. And so you have an increase of SF2, and the increase of SF2 binds to this to the enhancer and causes skipping of exon 11 and finished. Is my time finished?
Yeah.
Eh?
Yes.
Finished?
Yeah.
Oh, five more minutes. So this is the model that comes out. Are you sure it's finished?
Yeah.
No.
Yeah, it's 45 minutes.
Well, I haven't finished. Sorry. Now, I talked about the enhancer that binds SF2. Next to it, there is a silencer. That took much more time to find out what it is, what binds to the silencer. Turns out that we identified the silencer next to and it turns out that the silencer binds HNRMPA1. You already heard about it. It interacts with the silencer and it counteracts the activity of SF2. So A1 and SF2 are antagonistic only in this case. I don't show you all the evidence, but in the teaching material, there is the paper published on nucleic acid research a year ago that describes what happens. So the working, I have to skip everything because that's not the whole story. Because the whole story, it turns out so that HNRMPA1 does the opposite of what SF2 does. It induces the reverse process, mesenchymal to epithelial transition rather than, and it affects cell migration. In this case, instead of the migration through the membrane, is a wound healing example. So you take a layer of cells, you make a strip with a pipette point, and the cell, if they go through EMT, the wound heals, and it doesn't heal if A1 acts. Anyway, that's the other thing that was very funny is that A1 interferes with the production of another HNRNP, A1B2 called, that is a stimulator. Behaves like also through the NMD mechanism, and A1B2 behaves like SF2. So it's an enhancer. So I go to the. Is it sure that I thought I had an hour.
No.
No.
It's 5 days we work.
Okay, conclusions. HNRMP1 is a splicing repressor that interacts with the RON silencer and controls its activity. It also interacts with this other protein. So it modulates this other protein through AS-NMD. And this is the overall model of control of delta RON production. You have an enhancer where SF2 binds. It strengthens this site. But this site can be strengthened by also by this protein, by the mechanism which is not completely known. Okay? HNRMPA1, by binding to the silencer, displays the SF2 and also reduces the level of this other protein. And so you have either the empty process, epithelial to mesenchymal transition, if you have this situation, or the opposite process if A1 prevails. Okay? So it's a very complex network with a hierarchy of factors that in the end produce a very dramatic effect on cells. But before I finish, I want to mention that this work has mostly been done by Claudia Ghigna. Sorry, I don't have the picture. It's a nice girl. And Giuseppe Biamonti, and in collaboration with these guys in Trieste that you all know. And thank you very much. I thought I had 10 more minutes.